Hello, and thank you for watching Cardiovascular Physiology Part 3. As I have done with some of the previous lectures, I will present this lecture, this Part 3, in two segments. In segment one, I'll go through the first three topics here, and that corresponds with slide 1 through slide 58. And in the second segment, I'll pick up with point 4 here, hemostasis, and that's slide 58 to 105, the last slide. So we'll start by describing the composition and characteristics of blood. It's a pretty neat artistic representation of blood. Blood is a connective tissue, and it's one of the only, there are two liquid tissues in the body. Blood is one of them, semen is the other. So here's a phrase for describing the general structure, the generic structure of any connective tissue. Cells embedded in an extracellular matrix. Perhaps this phrase sounds familiar to you because in a previous class you studied bone tissue or cartilage tissue or some other connective tissue. It's a phrase for understanding the basic structure of any connective tissue. And the underlined portions of this phrase, cells and extracellular matrix, those are going to be specific, quite specific to the particular connective tissue that we're talking about. So all connective tissues can be viewed through this lens, cells embedded in an extracellular matrix, and we're going to apply this specifically to blood, where we'll talk about the different types of cells, and then we'll talk about the properties of the extracellular matrix that make up blood. So the ratio of cells to matrix is shown above. Blood is 45% cellular and 55% plasma, and plasma is the name of the extracellular matrix. There are three different types of cells in blood: red blood cells, white blood cells, and platelets. And collectively, blood cells are referred to as formed elements. So if I'm walking down the hall and Dr. Reyes says to me, "Hey Laura, what are you talking about in class today?" and I say formed elements, he knows we're talking about blood cells. And this is what blood looks like through a compound light microscope. So the way this slide was made is blood, the liquid, was smeared across a glass plate, and then a particular stain was added. It's called Wright's stain, W-R-I-G-H-T. And that stains the cells. Red blood cells are actually naturally red, but they take on sort of a pinkish appearance with Wright stains. And white blood cells are actually naturally white, but they take on purple tones using this Wright stain. So we can see the three different types of cells, the three different types of formed elements. The vast majority of the cells shown here are red blood cells. And then we see, I see 4 white blood cells. And there are several different types of white blood cells, but for now we're just talking about white blood cells in general. And then we see platelets. Whenever you see something that looks like schmutz on the slide, that's probably a platelet. And then, not really well indicated here, but the liquid that is surrounding these cells is called plasma. So this is an artificially colorized photomicrograph of the three types of formed elements: red blood cells, white blood cells, and platelets. Okay, so 45% of blood is cellular, 55% of blood is matrix, and it's a liquidy matrix. And that matrix is called plasma, and it's a body fluid, and it's mostly water, like all other body fluids are mostly water with a variety of dissolved solutes. We can use this diagram to illustrate the composition of blood. Blood makes up 8% of your body by mass. If we take whole blood and divide it into plasma and formed elements. So moving from the left side of the image toward the right, we see that 45% of your blood is cellular, those are the formed elements, and 55% of your blood is liquid, that's plasma. So if we take a closer look at the formed elements and we separate those by type, the vast majority of cells in your blood are red blood cells, there are white blood cells, and there are platelets. Eventually we'll talk about the function of each of these different cell types. For now, I'll just draw your attention to the white blood cells and ask you to continue looking forward, looking to the left, where we see 5 different types of white blood cells: the neutrophils, the lymphocytes, monocytes, eosinophils, and basophils. So each of those types of white blood cell has a particular function, but collectively they all participate in the immune system. All right, looking up top, going back to blood plasma, which makes up 55% of whole blood. Blood plasma is mostly water, 91.5% water, with solutes dissolved in it. 7% of whole blood of that plasma, is protein, and 1.5% is made up of other solutes. So let's go up to the top and look at proteins, moving over to the right. These are the 3 categories of plasma proteins: albumins, globulins, and fibrinogen. All of these proteins contribute to the osmotic pressure that's important during capillary exchange. Albumins up top, many of those proteins are chaperone proteins. They accompany lipid-soluble materials like lipid-soluble signaling molecules, travel in the blood associated with an albumin, a chaperone protein. Globulins are immune system proteins. Perhaps you've heard of antibodies. Antibodies are also known as immunoglobulins. So globulins are antibodies, and then there's fibrinogen. Fibrinogen is a precursor to the protein fibrin, which forms during blood clotting. So we'll talk more about fibrinogen when we get to mechanisms of hemostasis in the second segment. And then let's move our eyes to the other solutes that are part of blood plasma. These solutes include electrolytes. Electrolytes are substances that contribute to the generation of electrical signals. You know all about electrical signals now. So electrolytes include ions like sodium, potassium, chloride, magnesium, etc. Nutrients like glucose and amino acids, gases such as oxygen and carbon dioxide, regulatory substances. That's referring to signaling molecules like hormones, as well as enzymes, and then waste products like creatinine, like bilirubin, are all these other solutes. Okay, so here we have the general overview of the composition of plasma. I'm sorry, the composition of blood. So if we take a sample of blood and we put it through a centrifuge, it's very easy to separate the plasma from the cells. And within the cells, the red blood cells separate from the white blood cells and the platelets. So you've probably given a blood sample at your physician's lab. Hopefully it's just for a routine check, and often that blood is delivered into tubes that can easily fit into a centrifuge, because many different blood tests require that plasma be separated from the cells in order to perform the test. So let's take a closer look at your red blood cells. Red blood cells are also known as erythrocytes, where erythro means red and cyte means cell. And erythrocytes are basically bags of hemoglobin. They don't have any nuclei, they don't have any organelles. During the development of red blood cells, the nucleus and the organelles are ejected from the cell, essentially to make room for more hemoglobin and to allow the cell to take on this characteristic shape. It's called a biconcave disc. And this shape allows the cell to have greater flexibility. They're quite bendy. They can take steep corners. and they have a lot of surface area. The purpose of hemoglobin, and there are millions of molecules of hemoglobin inside each erythrocyte. The purpose of hemoglobin is to reversibly bind oxygen. So oxygen joins hemoglobin, oxygen will then dissociate from hemoglobin. And that oxygen needs to be delivered to all of our body tissues. And that oxygen is going to bind and then dissociate from hemoglobin, and it needs to cross the plasma membrane in order to do that. And so the biconcave disc gives the surface of the cell more surface area. So for oxygen to enter or leave the cell, that has to happen across the surface. And the more surface, the more plasma membrane there is, the more opportunity there is for oxygen to get into the cell or out of the cell. Here's an overview of white blood cells. They're also known as leukocytes, where leuko means white and cyte means cell. There are 5 different types of white blood cells in the body, and then even within those 5 types, there are subtypes. White blood cells are pretty complex. Each one of them has its own unique set of functions, but collectively, they are part of the immune system. So they help ward off disease. They help heal infections, for example. Certain types of white blood cells, like the neutrophils and the monocytes, they are able to leave the blood vessel. They can migrate out of the blood and they can join a body tissue if there's a pathogen that's been detected there. So in this image here, we're looking at a particular type of white blood cell called a monocyte. And monocytes can change their shape in very significant ways. So the cytoskeleton of the cell is responsible for determining the cell's shape. And so by manipulating its cytoskeleton, this cell can squeeze through the intercellular clefts in the wall of a capillary. It can leave the blood and enter a different tissue in order to fight a pathogen. Perhaps a bacterium is there, and the bacterium is sending out signals, and the monocyte, or the neutrophil in this case, the neutrophil is able to identify the bacterium by the chemical signals that it's sending out, and then presumably go and engulf that bacterium. Pus. We get to talk about all the good body fluids in here. So we've all experienced pus, this white, thick fluid that's associated with infection and the healing process in the human body. So pus is accumulated dead white blood cells plus bacteria plus tissue fluid. And of course here we're looking at a picture of a pimple or a whitehead. Platelets. Platelets are also known as thrombocytes, where thrombus means clot, and cyte means cell. So thrombocytes are not actually complete cells. Rather, they are cell fragments. During the formation of platelets that occurs in red bone marrow, it starts off as a cell called a megakaryocyte. So this very large cell that then fragments into hundreds of fragments that go on to become platelets. Platelets play several different roles. They are most well known for playing a role in hemostasis, and that means limiting blood flow out through the wall of a broken vessel. So they contribute to hemostasis, stopping blood flow. They also play a role in inflammation, which is an immune response following infection or injury. When platelets become activated, they undergo something known as the platelet release reaction, and they change shape very dramatically. So they go from being sort of these amorphous blob-like structures to becoming very spiky in appearance. And these radiating processes that they take on that we can see in this image help the platelets attach to each other. They get caught up on each other. Rather than easily slipping past each other, they get caught up on each other, and in this way they can contribute to forming what's known as a platelet plug. And we'll talk much more about that later. So we just looked at a few pictures to help reinforce these different cell types that occur in blood, these formed elements. And next, I'll try to reinforce the composition of plasma. So the most abundant solute in blood plasma is protein, and there are three different types or categories of protein that make up blood plasma. The albumins tend to act as chaperone proteins for fat-soluble signaling molecules and fatty acids. Fatty acids are actually a nutrient that needs to circulate in your blood. And then the globulins are immune system proteins. Perhaps you've heard of antibodies. They're also known as immunoglobulins. And as part of your immune system, antibodies or immunoglobulins are able to bind to different proteins that are part of the body of a pathogen, like a protein that's on the exterior surface of a particular bacterium or a particular virus. And then fibrinogen, that's just a single type of protein. Fibrinogen is produced in your liver, and it's an inactive protein, and as we will see, it's important for the formation of a blood clot. And we'll talk about that in the second segment as one of the mechanisms of hemostasis, limiting blood flow through the wall of a damaged blood vessel. So we'll talk more about fibrinogen later. And then the other solutes that we find in plasma are electrolytes, nutrients, gases, enzymes and hormones, and waste products. So what are examples of each of these substances? Electrolytes we've actually talked about quite a bit. An electrolyte is something that contributes to the generation of electrical signals in the body. So you know quite well the roles of sodium, potassium, calcium, chloride, etc. Those are all electrolytes. Nutrients like glucose and amino acids. Gases include oxygen and carbon dioxide. Enzymes and hormones, enzymes such as renin, which will become more important for us in the urinary system when we talk about the regulation of blood pressure and blood volume. And then hormones, a great variety of signaling molecules travel in our blood. Perhaps the one that we've talked about most so far is epinephrine and norepinephrine released by the adrenal medulla. And then waste products like bilirubin and creatinine, that's what it's called, bilirubin and creatinine. Creatinine is a waste product generated by our muscle cells, and bilirubin we'll talk more about in this chapter. Bilirubin is generated as a breakdown product from hemoglobin. Here are some of those examples listed out for you. All right, the physical characteristics of blood, it's actually thicker than water. In some places in the world, the expression blood is thicker than water is meant to indicate that family is more important than friends, for example. Blood is sticky. You've had experience with your own blood and perhaps it dried on your fingers and your fingers felt tacky. The pH of blood is very strictly regulated. It has this narrow normal range between 7.35 and 7.45, and please do memorize the pH of blood. And in your body, depending on your body size, there's somewhere between 4 and 6 liters of blood. And blood clots, it gets clumpy when it's outside the body. This is a very particular property of blood. And whenever we withdraw blood from the body, in order to prevent it from clotting or getting clumpy, we need to treat that blood with an anticoagulant. And one such anticoagulant is heparin. So perhaps the tubes at the bottom of this screen look more familiar to you because you've had blood drawn in a diagnostic lab. Hopefully it's just for routine testing. But some of these tubes that your blood is drawn into, some of these tubes are coated with heparin, and that's to prevent clotting, which will make it easier for them to do whatever testing they need to do on your blood. And then up top, those are capillary tubes. You can see how tiny they are, and they are coated, their interior is coated with heparin. I used to use those capillary tubes to collect blood samples from lizards and then put those little tubes, believe it or not, in a centrifuge so that I could separate the plasma out from the cells. And here's a great vocabulary word. Wherever you are, I'd like you to try to work this vocabulary word into the next meal that you share with someone or the next phone conversation that you have. And it's hematophagous invertebrates. And two of them are shown here. There's a mosquito and there are leeches. So there are lots of critters in this world that make their living by eating blood. And that's what hematophagous means. Heme means blood, phage means to eat. So these are both blood-eating organisms, and they're invertebrates, meaning they don't have an internal skeleton. They don't have a spine, for example. Mosquitoes are the most dangerous organism in the world to our species. All right, so the composition of your blood is strictly regulated, and there are many different organ systems that are constantly contributing to maintaining this pretty precise composition of blood. Your cardiovascular system, your respiratory system, urinary, endocrine, and nervous systems all play a role in maintaining these characteristics of blood. Your blood pH, your blood volume, how much blood there is in your body, your blood pressure, and your blood osmolarity. Hemoglobin. It's one of my favorite molecules. It's super cool, and I hope to convince you of that. So hemoglobin is described as a metalloprotein. It's a protein that includes a metal, and that metal is iron. And there are 4 different polypeptide chains folded together that make up the protein portion of the hemoglobin molecule. In this diagram here, we see that there are 2 beta polypeptide chains and 2 alpha polypeptide chains. So that's a total of 4 polypeptide chains. Like any protein, they're folded in a very specific way, and these 4 protein polypeptide chains are folded around each other. And as the protein folds, it incorporates 4 hemes into the molecule, which you can see there. There are 4 hemes. And in this image, we can take a closer look at the structure of the heme. So on the right side of the diagram, that's a heme, and at the core, the center of the heme, is an atom of iron. And iron is a metal, and it's this atom of iron to which molecular oxygen will bind. So this is how hemoglobin can reversibly bind with oxygen. That oxygen associates with the iron portion of the heme. So every molecule of hemoglobin has 4 hemes, and each heme can reversibly bind to a single molecule of O2. So this hemoglobin molecule that we see here can possibly carry up to 4 molecules of oxygen. And red blood cells are basically bags of hemoglobin, something on the order of 300 million molecules of hemoglobin inside each one of those cells. And remember, these cells don't have any nuclei and they don't have any other organelles. They are able to maintain characteristic gradients across the membrane. They do have sodium-potassium pumps, for example, and the way they generate ATP is by glycolysis through aerobic cellular respiration. Remember, they don't have any mitochondria, so they can't perform aerobic cellular respiration. So they must have a very low metabolic rate, and they do. They basically have to run those pumps, and that's about it. Okay, so as I mentioned earlier, hemoglobin is the oxygen carrying molecule inside our red blood cells, And when oxygen joins hemoglobin, when it binds to a heme, that's called oxygen loading. And when oxygen dissociates from the heme, from that molecule of hemoglobin, when it leaves the red blood cell, that's called oxygen unloading. And so in this diagram, we can see those processes. As we start on the left and move to the right, we see that this molecule of hemoglobin has gained 4 molecules of oxygen, and that's called oxygen loading. And as we go from right to left, this is oxygen unloading. So going back to left to right, oxygen loading occurs as blood travels through pulmonary capillaries. As blood passes through the capillaries in our lungs, it gains oxygen through a process known as oxygen loading. And then the reverse situation occurs as blood passes through our systemic tissues. As blood passes through the capillaries that are passing through a muscle or through a region of fat tissue, for example, oxygen dissociates from the heme and eventually leaves the red blood cell. And so in this diagram, we'd be going from right to left, and that process is called oxygen unloading, and that oxygen is left there at the tissue. It's taken up by our tissue cells. Hemoglobin also reversibly binds with carbon dioxide. And the carbon dioxide does not bind to the heme. Rather, carbon dioxide has an affinity for the amino acid portion of the hemoglobin molecule. And a significant portion of carbon dioxide that's in your blood right now is traveling in the form of what's known as carbaminohemoglobin. And I love that word. It's hard for me to say without smiling. Carbaminohemoglobin is hemoglobin that has carbon dioxide associated with it. And that carbon dioxide will dissociate from hemoglobin as that blood passes through your pulmonary capillaries. That carbon dioxide will leave the red blood cell. Carbon dioxide will unload as that blood passes through your pulmonary capillaries. All right, hemopoiesis and hemoglobin recycling. Hemopoiesis refers to the formation of new blood cells, all new blood cells. Red blood cells, white blood cells, and platelets, all of them are generated inside your red bone marrow. So perhaps you've studied the structure of bones before. Here we're looking at a long bone. It happens to be the humerus. And in the epiphyses of a long bone, that means the ends of a long bone, those ends internally, they have a lot of what's called spongy bone tissue, or sometimes it's called cancellous bone tissue. And inside the little nooks and crannies of that spongy bone, we find red bone marrow. And red bone marrow may also occur in the diaphysis, in the shaft of a long bone. And that's true when we're children, but as we move through childhood and into adulthood, we reduce the overall quantity of red bone marrow in our bodies. Much of that red bone marrow, particularly in the diaphyses, in the shafts of our long bones, like our humerus and our femurs and our tibia, for example. Much of that red bone marrow gets converted to yellow bone marrow as we become adults, and that yellow bone marrow is fat. But as adults, we need to retain red bone marrow so that we retain the capacity for hemopoiesis. We're always synthesizing new blood cells. So in this illustration, we can see the location of red bone marrow in the adult body. So adults have less red bone marrow than children do. And we find our stores of red bone marrow in the bones of our skull, in our ribs, in our pelvis, in the epiphyses of our long bones, the ends of our long bones. So hemopoiesis refers to the formation of any type of formed element. We're going to focus on erythropoiesis. Erythropoiesis is the formation of new red blood cells specifically. So because our red blood cells don't have any nuclei, they're not able to maintain homeostasis particularly well. They're brand new, they're born in red bone marrow, but then they're not able to maintain themselves. They can't synthesize new proteins. And so over the span of a few months, their plasma membranes degrade and they become leaky and worn out, and they get taken out of circulation. So we're always producing millions of red blood cells per second, which must also mean that we are removing millions of old red blood cells per second from circulation. White blood cells fluctuate in number over the course of days and weeks and months, whereas the concentration of red blood cells, you can think of it as a concentration, your hematocrit. Red blood cell concentration is more constant over time. White blood cell concentration can fluctuate dramatically, or your white blood cell count. And it depends on what's taxing the immune system. So all hemopoiesis begins with a particular category of stem cell that we find in red bone marrow, and they're called pluripotent stem cells. So pluri means a plurality. So these cells that we find in red bone marrow have the capacity to become a handful of different cell types in the body. They're not totipotent stem cells. Embryonic stem cells are totipotent, meaning they have the capacity to become any cell type in the human body. That's not what we find in red bone marrow. We find pluripotent stem cells. They have a plurality of potentials. Please don't be scared by this diagram. So up top, we have one of these pluripotent stem cells, and it will go down a particular developmental pathway. It will differentiate into one of the cell types shown below. And how this cell differentiates is by cell signaling. In its environment, it receives different signaling messages released by neighboring cells and released by other cells distant in the body, like kidney cells, for example, release a hormone called erythropoietin. Erythropoietin increases the production of red blood cells specifically. So these signaling molecules that are in the environment of a pluripotent stem cell determine if that pluripotent stem cell is going to go on to develop into a myeloid stem cell, shown on the left, or a lymphoid stem cell, shown on the right. And on the right, the lymphoid stem cells give rise to T cells, B cells, and natural killer cells. If you've studied the immune system before, perhaps you recall that one of the types of white blood cells are the lymphocytes. And then within the lymphocytes, there are 3 different types: T cells, B cells, and natural killer cells. And even within those, there are subtypes. And if this pluripotent stem cell does not become a lymphoid stem cell, it becomes a myeloid stem cell. So looking at the left here, that myeloid stem cell could then go on to become a red blood cell, platelets, or the other types of white blood cells: eosinophils, basophils, monocytes, or neutrophils. So please don't memorize this diagram. Please do know pluripotent stem cell. Do know that cell signaling factors determine the differentiation pathway that that cell will undergo, and that a pluripotent stem cell has the capacity to differentiate into any of the different formed elements we find in blood. And here's that flow chart written out. If we start out with a pluripotent stem cell, it's initially destined along one of these 2 paths. It's either a myeloid stem cell or a lymphoid stem cell. Myeloid stem cells have the capacity to become red blood cells, platelets, or these other types of white blood cells: the eosinophils, the basophils, neutrophils, and monocytes. And then if that pluripotent stem cell becomes a lymphoid stem cell, that's going to give rise to some type of lymphocyte, a natural killer cell, a B cell, or a T cell. So bone marrow cells are a source of stem cells, and stem cell research is a ripe area right now. We're developing fantastic therapies for treating burn victims, for treating spinal cord injuries, and other types of disorders and diseases using the patient's own cells. And the reason why we want to use a patient's own cells is that they are less likely to trigger an immune response. So someone is less likely to reject their own cells than they are to reject cells from a donor individual. And so we can harvest these bone marrow cells and subject them to a variety of external signaling molecules and cause these pluripotent stem cells to turn into different types of cells, like skin cells or cardiac muscle cells, for example. It turns out that these pluripotent stem cells that are part of red bone marrow, they have capacity beyond blood cells, meaning they can become several other types of cells in the body. They're not just limited to any of the formed elements. And one neat early piece of research that gave us, the body of science, the first clue that these pluripotent stem cells had more flexibility than just being able to become the formed elements came from a study that involved men who had received donor hearts. So they were all heart transplant recipients, and each of these men went on to finish his life. And when he died, his heart, his donated heart, was karyotyped. So many of the cells in that heart were examined to determine which chromosomes were present. That's what a karyotype is. And for 7 to 16, depending on the patient, here's the range, 7 to 16% of those heart cells contained a Y chromosome. So these men all received hearts that were donated by females. So our expected outcome for when the male heart transplant recipient passed away, and we looked at the cells in that donated heart, we expect 100% of them to have 2X chromosomes. That would be the result of their karyotype. And 2X chromosomes represents a genetic female. And remember, that heart originated from a female body. But that's not what we found. Instead, we found, let's say 16% of those heart cells had an XY karyotype. So where did the male cells come from? They had to originate somewhere in the patient's own body in order to have that Y chromosome. And so now that led us to search, what's the origin? What are those stem cells that are becoming cardiac myocytes? And with many other research papers, we now understand that these pluripotent stem cells in our bone marrow have the capacity to differentiate and join cardiac myocytes in the heart. Red blood cell homeostasis. As I mentioned earlier, our red blood cells have virtually no capacity to regenerate themselves, to repair their plasma membrane. So they have this finite lifespan. And as their plasma membrane starts to degenerate, they become leaky and they get taken out of circulation. They're taken out of circulation by phagocytes, macrophages in your liver, as well as in your spleen and other tissues. And these macrophages will engulf an old, worn out blood cell and will essentially recycle it for parts. There are some good, useful building blocks that make up the body of a red blood cell, and we certainly don't want to waste them. So if we're constantly taking red blood cells out of circulation, we need to constantly be adding red blood cells to circulation. So if your hematocrit is to remain stable, red blood cell formation has to equal red blood cell destruction. And so there are several different types of signaling molecule that influence red blood cell production, or erythropoiesis. Collectively, they're known as hemopoietic growth factors. And the one that we've talked about a little bit before, and I'll mention again here, is erythropoietin, or EPO. EPO is secreted by particular cells in your kidney, and those cells respond to hypoxia. When those cells are not receiving enough oxygen, they secrete erythropoietin, a hormone, into the blood. And that hormone acts on red bone marrow and initiates greater production of red blood cells. So when we increase hematocrit, when we add new red blood cells to the blood, that's going to reduce the incidence of hypoxia. So more oxygen will be delivered everywhere in the body, and particularly to those kidney cells. Those kidney cells will no longer be facing hypoxic conditions, and they'll stop secreting erythropoietin. And so this is a negative feedback homeostatic mechanism. We can use this image to reinforce that. So starting up top, we see decreased oxygen delivery to the tissues. That's hypoxia. There are oxygen sensitive cells in your kidney that detect that, and in response to hypoxia, they'll secrete erythropoietin. And erythropoietin is a signaling molecule. It travels in your blood. Bone marrow, red bone marrow cells have receptors for erythropoietin, and in response to erythropoietin, those red bone marrow cells change their behavior. They go down a particular differentiation pathway, and they become brand new little baby red blood cells. And so erythropoietin has the effect of increasing the percentage of red blood cells in your blood. It increases your hematocrit. And with an increasing hematocrit, we increase the capacity of your blood to deliver oxygen to your tissues. So we decrease the hypoxia. And so this negative feedback system will be shut off. So we're covering the hard questions today. Why is blood red, bile green, urine yellow, and feces brown? Why are all these substances very particular colors? The answer to every question here is hemoglobin, and we're in a position to understand that. So when old, worn out red blood cells are phagocytized, when they're engulfed by a macrophage, that cell is broken down, specifically the hemoglobin is broken down and recycled for parts. Some portions of the hemoglobin molecule are very valuable and can be reused, and other portions of that hemoglobin molecule are actual waste products that don't have any value and need to be eliminated from the body. So that arrow, that circle of arrows that we're seeing down below, that's what's known as closed loop recycling. When all of the molecules of that material stay in the loop, that's not exactly what happens for hemoglobin. We have open loop recycling that occurs with hemoglobin. So some of the molecular building blocks of hemoglobin are useful. They still have value, and they'll continue around the circle. But some portion of the hemoglobin molecule is a legitimate waste product that can't be reused, and that will need to be eliminated from the body as waste. So let's revisit the structure of hemoglobin. This is a different artistic representation, and I can use it to remind you that hemoglobin is a metalloprotein. There are 4 different polypeptide chains, and of course, those are made out of amino acids. And amino acids are building blocks, and they can be used to build proteins, many different proteins. Those proteins can be assembled, those proteins can be chopped up into amino acids, and then those amino acids can be used to build different proteins. So those polypeptide chains are going to be catabolized into amino acids, and those amino acids are good and useful, and they can be used to make ATP, or they can be used to synthesize new proteins. The heme. The heme includes some useful parts and some not so useful parts. So the iron component of the heme is a valuable substance. In some environments, iron can be a limiting nutrient. So if we think about humans and most of their evolutionary history, so 200,000 plus years, in certain environments, Nutrition-wise, iron wasn't particularly abundant, and we need iron. We need iron to make hemoglobin. And so iron is not wasted. And so that iron will be useful, as we'll see in a moment. But the heme, the rest of the heme, so we pluck out that iron and we're left with the heme pigment. That heme pigment is not useful, and that's going to become a waste product known as bilirubin, and it also has many other forms, but bilirubin is the form of that waste product that we'll talk about the most. All right, so how do we recycle hemoglobin? We're going to catabolize those polypeptide chains, so proteases, enzymes that digest proteins, will chop up those polypeptide chains into amino acids, and those amino acids are good and useful, and they will be reused. The iron is also good and useful. The iron gets stripped away from the rest of the heme molecule, and that iron is either going to be sent to red bone marrow or to the liver. If it gets sent to red bone marrow, that iron is going to be reused for the production of new hemoglobin molecules. And if it gets sent to the liver, that iron is going to be put into storage, where sometime in the future it might be used to synthesize new molecules of hemoglobin. And then that leftover portion of the heme with the iron removed, that gets degraded to a substance called bilirubin. And bilirubin is what's responsible ultimately for this greenish color of bile, for the yellow color of urine, and for the brown color of feces. It's a little more complicated than that. All right. I love this diagram. There's a lot going on here, but believe it or not, it's relatively simple to understand. So let's start with the pink blob that's on the left side of this diagram. That pink blob is a macrophage. So it's a cell that is stationed in your liver. Maybe it's a Kupffer cell. Perhaps you've studied the liver before. Or it's stationed in your spleen, for example, and its job is to identify and engulf cellular debris, so pieces of cells that have died, as well as leaky, worn out red blood cells. So that macrophage will engulf red blood cells, and then inside the macrophage, the hemoglobin recycling occurs, or at least it's initiated. So let's start with globin. Up top in blue, we're still inside the macrophage. Globin refers to the polypeptide, the protein portion of the hemoglobin molecule. And so the globin portion of the molecule is catabolized into amino acids. And those amino acids are released from the cell. They re-enter the blood, and amino acids are quite fungible. They can be used for synthesizing new proteins, different proteins, or they can be used for making ATP. That's the easy part. Let's take a look at the heme. The heme, you'll recall, is that iron-containing portion of the hemoglobin molecule, and that iron gets stripped away. And that iron gets released into the blood, and it is transported by a chaperone protein called transferrin. So now we're looking sort of in the center of this diagram. We see an atom of iron attached to a chaperone protein called transferrin. And the iron associated with transferrin travels in blood to the liver. It gets taken up by liver cells, and that liver can either store the iron there, so that's the end of the line for the iron, at least temporarily, it gets put into storage, or the liver can send that iron to red bone marrow. And if it ends up in red bone marrow, that iron will be reused to synthesize new molecules of hemoglobin. And iron is never left alone in the body. It's a limiting nutrient for many bacteria. And so if iron was not accompanied by a chaperone protein, it would become available to bacteria, and the bacteria could end up growing very, very quickly and causing serious infection. So iron is always chaperoned. It's never left alone in the body. Okay, going back into the macrophage, the big pink blob. Once we strip the iron from the heme, we're left with a greenish pigment called biliverdin, and that biliverdin gets converted into a yellowish pigment called bilirubin. And that bilirubin leaves the macrophage and becomes part of blood. There's bilirubin in your blood right now. And that bilirubin gets taken up by your liver. And your liver cells, your hepatocytes, one of their 8 million different jobs is to produce bile. We've not yet covered the digestive system, but bile is an important body fluid. It's produced by your liver, it's stored in your gallbladder, and it's secreted into your small intestine, where it aids in digestion. Particularly bilirubin or bile is an emulsifier. And we'll talk more about that when we get to the digestive system. So this bilirubin, as part of bile, gets secreted into your small intestine. And in your small intestine, as part of bile, it makes its way from your small intestine to your large intestine. In your large intestine, this bilirubin encounters bacteria. There are bacteria in your large intestine, and these bacteria are able to convert bilirubin into something known as urobilinogen. It still has this yellowish color. And it turns out that that urobilinogen can cross the wall of the large intestine and get back into the blood. And so there's urobilinogen in your blood right now, along with bilirubin. And these things can be filtered out by your kidneys and incorporated into urine. And so if we do an analysis of the composition of urine, we find bilirubin, we find urobilin, and those things contribute to the yellow pigment of urine. Moving along your large intestine, so going from number 12, I guess, to number 14, the urobilinogen that remains in your large intestine can be further modified by bacteria. And bacteria convert that urobilinogen into a different compound called stercobilin. And stercobilin has a brown color. And this is the reason why feces are brown. So maybe today you eat bananas, you eat strawberries, you eat papaya, and you eat spinach, and these things all have very different colors. But feces come out brown almost always, and this is due to the presence of stercobilin. And stercobilin is one of the breakdown products from bilirubin, and it's one of the ways in which your body excretes bilirubin, gets rid of it as a waste. Jaundice is described as a yellowish tinge to the skin, to the nail beds, to the eyes. And jaundice indicates, it's a clinical indicator of the accumulation of bilirubin in the blood. So normally there is bilirubin in our blood. It's always being added to our blood as hemoglobin is being broken down. Bilirubin is a breakdown product of hemoglobin. Normally, this bilirubin that's in our blood is constantly being collected by our livers and filtered out by our kidneys. Our livers incorporate bilirubin into bile, and bile is excreted. It leaves in our feces in the form of stercobilin, or it gets filtered out of our blood in the form of urobilin, as we covered in the previous slide. So in somebody who does not exhibit jaundice, there is bilirubin in the blood. It's constantly being added to the blood by these macrophages that are breaking down hemoglobin, and it's constantly being removed from the blood, primarily by your liver, and your liver incorporates it into bile. So when somebody shows jaundice, it indicates that there's too much bilirubin in the blood. And the most common cause of jaundice is a problem with the liver. For some reason, those liver cells, those hepatocytes, are not adequately removing bilirubin from the blood. And that means that bilirubin is able to accumulate in the blood. And we can see this because bilirubin has a yellowish tinge to it. Perhaps you vomited bile. I hope you haven't, but so many of us have had this experience. you vomited, and then you vomited again, and then you vomited a third or perhaps a fourth or a fifth time, and there's definitely nothing, there's no food material remaining in your stomach. And then you vomit up this yellow, frothy, very bitter liquid. That contains bile, and that's why it's yellow. And bile includes bilirubin. And bilirubin in the blood is responsible for the yellowishness that we observe during jaundice. Jaundice is an indicator of liver dysfunction, and you should be seen by a healthcare professional quickly. All right. So this is the end of the first segment of cardiovascular physiology, part 3. I'll stop here, and where you'll pick up next is the second segment of cardiovascular physiology, part 3, where we'll talk about the mechanisms by which blood flow through a broken blood vessel is limited. How do you stop bleeding, essentially? And that's called hemostasis. See you there.